Hey guys, welcome back to another lecture video for Chem 104. In this lecture video, we will revisit this concept of oxidation and reduction now that we've discussed alcohols, aldehydes, and ketones. And so I personally feel that this slide is a good summary slide of all the different transformations that occurs among aldehydes, alcohols, ketones, and carboxylic acids. And so if you guys recall, oxidation in general chemistry translates to a loss of an electron. And we're taking that definition and applying it a little bit differently in organic chemistry. And so an oxidation process involves a loss of a hydrogen. In this case, sometimes two, sometimes one, depending on the functional group. And when those hydrogens are lost, it enables the other element, in this case the carbon, the central carbon that you guys see on the screen, to make new connections to that of oxygen. So let's go ahead and take a look at this primary alcohol. And so if you guys compare and contrast the structure before oxidation occurred and the structure after oxidation occurred, notice that there's going to be a connection, another connection. So we're increasing the connection between carbon and oxygen. And I'm going to zoom in. Let's see here, let me erase that so it doesn't get too messy. Now, if you guys are looking at the number of hydrogens that is present on the alcohol, the primary alcohol, if you compare that to the number of hydrogens that's on the carbonyl carbon of the aldehyde, notice that we have a reduction in hydrogen. If you guys are looking at the carbonyl oxygen, notice that there are no hydrogens left over. So in the first oxidation step, so if we're going from a primary alcohol to an aldehyde, we're going to lose two hydrogens, and it's going to go somewhere. It's probably going to go on the catalyst, or it's going to be transferred to another compound. We don't really care where that hydrogen goes. All we know is that those two hydrogens are lost, enabling the electrons to go to carbon and oxygen, forming that second bond, that double bond. All right, and so here we lost two hydrogens, but we're gaining one connection between a carbon and an oxygen. And so here we have a double bond, here we have a single bond. All right. Now, if we're looking at aldehydes and carboxylic acids, I want you guys to notice the same idea, number of hydrogens, the number of bonds between carbon and oxygen. And so here I'm going from one, two bonds to between carbon and oxygen to 1, 2, 3 bonds between carbon and oxygen. And so I'm still gaining one carbon-oxygen bond if we're going in the forward direction, describing oxidation. At the same time, notice that the connection between this carbon and hydrogen is gone. And so we're losing one hydrogen when we're going from aldehyde into a carboxylic acid. Now, if you guys are looking in the reverse process is simply the reverse of what you guys see here. And so if we're going from a carboxylic acid to an aldehyde, well, if we reduce, so this is reduction, going backwards is reduction. If we undergo reduction, then here we're going to lose a connection between carbon and we're going to lose one connection between carbon and oxygen, and we're going to gain one connection to hydrogen. And so we're going to break this apart and we're going to replace this OH with the hydrogen if we're going in reverse. And so in a similar manner, if we're going from an aldehyde back to a primary alcohol, so if we're going in reverse, then we simply take the opposite of these values here. And so what we're going to get is we're going to lose one connection between carbon and oxygen, and then we're going to gain two connections to hydrogen. And so that's only going from an aldehyde to a primary alcohol. So if we look at a secondary alcohol, maybe I need to erase this. I'm going to erase this part. All right, so if we focus specifically on the secondary alcohol, if we go in the forward direction, which is oxidation, and if you compare these structures before and after, you guys will see that two hydrogens are lost. So we're going to take away two hydrogens. And notice that we get a connection, we gain one connection, we gain an extra bond between carbon and oxygen. So we're going to add one bond between carbon and oxygen. So it's very similar to the first oxidation from primary alcohol to an aldehyde. The process is the same. So we're losing the same number. I'm going to erase the reduction part so there's no confusion. So notice that for the oxidation of a primary alcohol and the oxidation of a secondary alcohol, in both scenarios, we're increasing the number of bonds between carbon and oxygen. And in both scenarios, we're losing two hydrogens. And so if we have a ketone, it is impossible for it to undergo oxidation simply because there are no hydrogens left on the ketone. So there's no hydrogen left on the carbonyl carbon. So if there's no hydrogens left over in the carbonyl carbon, then it cannot be oxidized. And so therefore, there's no product. When you guys are looking at a tertiary alcohol, same idea as the ketone. If you look at the carbonyl carbon, there are no hydrogens. And so therefore, this tertiary alcohol cannot be oxidized. Okay. And so that's pretty much a summary of the oxidation and reduction process. And so the next few slides specifically talks about reduction. It goes over the same type of descriptions that I've already discussed in this previous slide, but we'll just go ahead and go through it. And so this just shows the general mechanism of a reduction process that's occurring between a carbonyl group to produce some type of alcohol. And so we're generalizing this as a carbonyl. We don't know if it's an aldehyde or a ketone. But in both scenarios, when we reduce a carbonyl group, we're going to add two hydrogens. And so overall, we're going to lose one carbon-oxygen bond, and we're going to gain a bond between the carbonyl carbon and that of hydrogen. So all of that was described when we're going from aldehydes back to primary alcohols or ketones back to secondary alcohols. Okay. And so this just kind of demonstrates, this slide demonstrates an aldehyde being reduced to a primary alcohol. And so whenever you have an aldehyde and you reduce it, you create a primary alcohol. So this is a link that you guys should memorize. Aldehydes reduce, it creates a primary alcohol. Primary alcohol, oxidize, then that produces an aldehyde. And so one example of a reducing agent is something that we've already seen in chapter 11, which is hydrogen gas and palladium. So palladium is going to serve as our catalyst. And so if you guys have H2PD, this is demonstrating a reduction reaction. And so if we have this 2-carbon aldehyde, and so if you guys recall, a 2-carbon aldehyde is acetaldehyde. And if we reduce acetaldehyde using H2 gas and palladium as a catalyst, then we're going to get a primary alcohol. And if you check out the name of this primary alcohol, this is a 1,2-carbon alcohol. So this is ethanol. And so this is a reduction process that can occur in both in your body and in industry, right? And so once again, the presence of palladium and hydrogen gas is a common reagent to perform reduction. And so for ketones, so instead of an aldehyde, we have a ketone. So if you're starting off with a ketone, and if you guys are reducing that ketone, you're going to create a secondary alcohol. And so reduction produces a secondary alcohol. If you guys have a secondary alcohol, and if you oxidize it, you guys are going to get a ketone. And so this reagent, this is a reducing reagent, so we would not use H2PD to perform oxidation. H2PD is just going to be used for reduction. All right. And so why is this so important to us? And so the formation of double bond and single bond, this idea of a redox reaction, is actually really necessary for night vision. And so inside a human eye, there's 2 types of light-sensitive cells. They're called the rods and the cone cells. And so the chemistry of vision in the rod cells it kind of like centers around this aldehyde, this 11-cis retinal. So I'm just going to circle that real quick. And so you guys need this substance for vision, for night vision. And the aldehyde that we're referring to is circled in blue. Okay? So remember that aldehydes are terminal functional groups. And so you guys can imagine that, if your eyes are bombarded with light, and these light-sensitive cells, these rods, are converting your 11-cis retinol into 11-cis-retinal so that night vision can occur, then at some point in time you guys are going to consume all of 11-cis-retinol and conversely all of the trans-retinol. And so if you consume all of your cis-retinol and everything is converted into cis-retinal, if it's not replenished, then there's no specific chemical signal that will send a nerve impulse to the brain to convert that to a visual image. So in other words, there would be you would be night blind. And so for your rod cells to continuously communicate to the brain to process a specific image at night, your 11-cis-retinal needs to be converted. It needs to be converted back, and so this 11-cis-retinal is going to be converted into an all trans-retinal. So we still have the aldehyde. However, if you guys look at the double bond, specifically the relationship between the hydrogens on that double bond, it's going to be trans. Whereas if you guys are looking at the hydrogens here for 11-cis-retinal, these hydrogens are cis with respect to each other, and these hydrogens are trans with respect to the double bond. And so what's going to happen is your 11-cis-retinal is going to be consumed, and it's going to produce an all trans retinal. And so for you to replenish the 11-cis-retinal, which is needed for night vision, you're going to take the product, which is the all trans retinal, and you're going to convert it back into 11-cis-retinal. And this is done through 2 steps. And the steps is an oxidation reduction reaction. And so what's going to happen is that your all trans retinal is going to undergo reduction, and the reducing agent in this case is NADH. So if you guys have taken biology, you guys should have come across NADH when you guys were talking about, for example, ATP or the production of energy and all that stuff. And so NADH is going to act as a reducing agent, and it's going to convert this aldehyde into a primary alcohol. And so some enzyme is going to reorganize the isomerization, so it's going to convert the trans back into the cis. And so for the cis-retinol to become cis-retinal, which is needed for vision, this primary alcohol needs to undergo oxidation to reproduce the aldehyde. And the oxidizing agent is going to be NAD+. Okay? And so this cyclical motion of oxidation and reduction, there's biological relevance, and this is only one example, right, out of a lot of examples. And so without this oxidation reduction process, we wouldn't be able to see anything at night. All right. Cool. So the next thing that I want to discuss is being able to test for aldehydes and ketones. And so there's very specific tests that I want to talk about. The first one is called the Tollens Test. And so the Tollens Test, so the idea is that you guys have like this unknown solution. And so let's just say that somebody gave you this unknown solution and said, hey, you know what? There's a functional group in that solution. I don't know what it is. Can you run some reactions to inform me what type of functional groups I have? And you're probably going to be like, okay, sure, I'm an organic chemist, I can do this. And so one of these tests, it's called the Tollens test. So this Tollens test is going to check for the presence of aldehydes. Um, and so the main mechanism is that you have this aldehyde. So if your mysterious solution just happens to contain an aldehyde, so if you mix this aldehyde with Tollens reagent, and so Tollens reagent is specifically this, an oxidation reaction will occur such that this aldehyde is going to be converted into a carboxylic acid. Okay, so oxidation means that you're losing hydrogens, right? So we're going to go ahead and break that, destroy that hydrogens. It goes somewhere. We don't really care. And what we get in its place is a hydroxyl group from a water molecule that came from somewhere. We're not going to be focused on the mechanism, but altogether you just get a carboxylic acid. And as that electron transfer, that oxidation reduction reaction occurs, this silver that's found in your Tollens reagent, it's going to go from aqueous to something solid. And so what you get is this very it is silver. It's a silver mirror, and so you get this product it looks pretty cool, and so you get pure silver that's solid in state. It's very reflective. Okay. And so here your aldehyde is not constrained to a chain. Your aldehyde can also be a ring, a cyclohexane, for instance. And so here, if you have this aldehyde and if you treat it with Tollens reagent, then it's going to oxidize the aldehyde so that it can become a carboxylic acid. And a byproduct of that oxidation reaction is producing this solid silver. Okay. And so if you were presented with an unknown solution, and if you use Tollens test and you get this silver looking structure in your test tube, then it's positive for aldehydes. Now, if you guys don't get a reflective silver mirror, then it just tells you that an aldehyde is not present. It doesn't tell you that a ketone is present. It could be like, for example, an alcohol, right? And so a false result will tell you that an aldehyde is not present. It could be something else. And so another test that I want to talk about is called the Benedict's test. And we'll see it in chapter 13 when we talk about carbohydrates. And so Benedict's test, what it does is that it tests for the presence of aldehydes, but not simple aldehydes. It has to be, the arrangement is very specific. So let me just go ahead and draw your attention to this image down below. And so if you guys have a organic compound such that you have an aldehyde, and then right next to the aldehyde, one carbon away, there's an alcohol. This combination will allow this copper to be reduced into copper 2 oxide. So this copper 2 oxide is going to turn brick red. And what you get is the oxidized form of that aldehyde, which is going to be a carboxylic acid. And so if you only have an aldehyde and there's no alcohol, one carbon away, then it will result in a negative test. So it's going to produce a negative result for Benedict's test. Okay. So this is, we'll talk about this again when we talk about carbohydrates in the next chapter. But the prerequisite is that we have this aldehyde, and one carbon away, exactly one carbon away, there's an alcohol. If that condition has been met, we can treat it with Benedict's reagent, allowing an oxidation reduction reaction to occur. Basically, the copper 2 will get reduced to copper 2 oxide, and the aldehyde gets oxidized to become a carboxylic acid. The OH in itself doesn't change, but it is necessary for it to be there. It allows the purpose of the hydroxyl group is supposed to act like as an anchor to Benedict's reagent. So we're not going to go ahead and we're not going to talk about the exact mechanism, but I'm just going to go ahead and throw it out there that hydroxyl group is required. And so what does this look like? And so Benedict's reagent in its reduced form, when in its oxidized form, that means there's no electrons at all, it's going to be blue. However, when copper becomes reduced, it means it gains electrons, it's going to turn into a brick red color. So if we were to do this in lab, we can go ahead and describe a positive or negative result based on the color that we observe. And so this is typically applied for carbohydrates. And so here carbohydrates, there's a class of carbohydrates that has aldehydes, and there's a class of carbohydrates that has ketones. And so one way to use the Benedict's test is to identify if you have an aldose or a ketose, and we'll talk about that in the next chapter as well. But if you guys look, these carbohydrates, they have that hydroxyl group on the carbon that's right next to the aldehyde. And so after the oxidation occurs, then what we get at the very end is a carboxylic acid. And the visual conformation is the change from blue to red. Okay. And so what we're going to do is we are going to go through some examples, and we're going to predict the product of oxidation and reduction for these reactants. And so since this slide is focused on oxidation, we're going to go ahead and put in a bracket with an O to describe that an oxidizing reagent is required. So one example of an oxidizing agent is potassium permanganate. There's so many more oxidizing agents out there. But not all oxidizing agents will react with the secondary alcohol. So we're just going to broaden it. Excuse me. So we're just going to go ahead and broaden the idea of an oxidizing agent by putting this bracket in a letter O. And so looking at this alcohol, notice that this is a secondary alcohol. This central carbon has two carbons attached to it. And so if you have a secondary alcohol, what product do you get? And so if you guys don't recall, just think about what oxidation means. So oxidation means a loss of a hydrogen, right? And so if you're trying to take away a hydrogen, that electron has to go somewhere, so you have to gain a connection with, we're going to gain a bond between carbon and oxygen. And so the result that we're going to get is going to be a double bond to this oxygen. So we get a ketone. And you can go ahead and name this ketone by saying 1, 2, 3, 4, 5, 6, 7. So this is going to be 2-heptanone. Okay. Whereas the name for this substance, this reactant, is going to be 2-heptanol. All right. So let's go ahead and go through another example. Once again, we're going to go ahead and communicate to the reader that an oxidation reaction is going to occur. And for us to identify the product, we're going to have to think about whether or not this is a primary alcohol or secondary alcohol or tertiary alcohol. And so let's look at our carbons. So this carbon is connected to one other carbon. And so since that carbon is connected to one other carbon, then this is going to be a primary alcohol. And primary alcohols, when it undergoes oxidation, it's going to lose, 2 hydrogens, and it's still going to gain one connection between carbon and oxygen. And so if I draw out all of my hydrogens that's on that carbon, these 2 hydrogens are going to be lost somewhere somehow. And what that's going to do is going to leave me with an extra electron on the oxygen and an extra electron on the carbon, and these two can form a bond. And so the final structure that I'll get is this. Okay. So notice that this is, this is an aldehyde. All right, and so if we want to name this aldehyde 1, 2, 3, 4, 5, and so this is going to be pentanal, and then we have a methyl group on the third carbon, so this is 3-methyl. Pentanal. Remember, we don't use the carbon number 1. We're going to omit 1 in the name because aldehydes are always terminal functional groups. All right, so let's go ahead and look at the next one. Here we have this alcohol once again, and for when it undergoes oxidation, it's going to produce a different functional group. So we need to figure out what functional group it'll turn to. Well, to do that, we're going to go ahead and look at the carbons that it's connected to. So it's connected to 2 carbons. So therefore, this carbon is going to be a secondary carbon, making this alcohol a secondary alcohol. And so if you have a alcohol that's in the middle of a chain, another way for you guys to think about this, if you have an alcohol that's in the middle of the chain and if you're oxidizing it, remember you're trying to lose a hydrogen. So if you lose that hydrogen and we're trying to gain one bond between carbon and that oxygen, then what we get is a ketone. Okay. And so that would be my final answer. So it sounds really complicated at first, these predicting the products for oxidation reduction reactions, but really there is a trend. I mean, we only have it's almost like a 50-50 shot. Aldehydes are ketones from alcohols. And so if you have a primary alcohol, you get an aldehyde. Primary alcohols are always on the terminal end of the carbon. It makes sense for it to produce a terminal functional group, which in this case is the aldehyde. If you have a secondary alcohol, that alcohol is in the middle of a carbon chain, and for now the only functional group that you know of that'll be found in the middle of a chain is a ketone. And so we'll get a ketone wherever that alcohol is at. All right, so let's go ahead and predict the products once again for these three. And so for this molecule, if an oxidation reaction occurs, this hydroxyl is in the middle of a chain. So if it's in the middle of a chain, if it undergoes an oxidation, we're increasing the number of connections between carbon and oxygen and decreasing the number of connections between carbon and hydrogen, or in this case, oxygen and hydrogen. And so therefore, what we're going to get is a ketone at the very end. So for this next example, I'm going to go ahead and look at my carbon and the alcohol that's attached to it. Here I have the carbon that's in blue is connected to 1, 2, 3 carbons. And so this is a tertiary carbon. If we introduce it to an oxidizing agent, there's no reaction that's going to occur. And so here, last but not least, we have this carbon. This carbon is connected to 1, 2 carbons. So this is a secondary carbon. And if we oxidize a secondary carbon, it's going to produce a ketone. Oh, it's actually the same one. Oops, oh well. All right, so hopefully you guys kind of understand that logic. So let's go ahead and talk about reduction. And this time, we're going to specify our reducing agent to H2PD. And so here we have a ketone. If we're going to reduce it, right, so that's why it's called reduction, then that means we are going to add hydrogens to where that carbon, to where that carbonyl group is. And the resulting structure that we're going to get is an alcohol. Okay. And so how does an alcohol form from this carbonyl, from this ketone? Well, one of those hydrogens is going to be connected to this oxygen, and the other hydrogen is going to be connected to this carbon. Okay. And so if you guys look at this carbon right here, this carbon has 1, 2, 3, 4 bonds. And so it has no hydrogens to begin with. However, when you guys are looking at the product, here we have 1, 2, 3 bonds. And so that fourth bond, it's implied that there's a hydrogen that was added to it, right? So that was this hydrogen right here. And so when the reduction reaction occurs, this bond is going to be destroyed, and these bonds, these dotted lines, become permanent bonds. And we're also going to lose one connection between carbon and oxygen. So long story short, if you guys are getting overwhelmed with the mechanism, you guys can just do this very simply. Ketone produces secondary alcohol. And as long as you guys remember that, then that's really all that matters. All right, so here we have another ketone. Notice that it's in the middle. And so if we treat this with H2PD, what we're going to get is an organic compound. Looks kind of funky right now. And so this is where that ketone was located. And wherever that ketone is located, we're just going to replace it with an alcohol. So once again, that H2, those two hydrogens, one is going to connect to the oxygen, the other will connect to the carbon. Okay, you guys can think about it that way. And so put this together, you guys create your alcohol. And the hydrogen that was added, the second hydrogen that was added, is invisible since we're using the skeletal structure. And so once again, we get a secondary alcohol. All right, and so let's go ahead and reduce this substance. Notice that here we have a secondary carbon. This is a ketone. And so if we're going from a ketone, and if we're reducing it, then we should get a secondary alcohol. Now, the reason why I chose this example is because there's a double bond. And so if my reagent is H2PD, if you guys recall from chapter 11, what happens when you have a double bond and it's being introduced with H2PD? And so it will convert that double bond into a single bond. So my final answer is just going to be this carbon chain with no double bonds, and everything is converted to alcohol. All right. And so I'm just going to do a couple more here. Maybe I'll just do the last two just to demonstrate the point. And so here, if I treat this with H2PD, if I look at my functional group, I have an aldehyde. And so if I have an aldehyde and if I'm trying to reduce it, the product that I'm going to get is a primary alcohol. And so I'm just going to go ahead and copy the parent chain down exactly as is. And wherever that aldehyde was, which is on the terminal point on the left, I'm going to go ahead and replace it with an alcohol or a hydroxyl group. And so just to demonstrate the mechanism very quickly, this H2 gas, so I'm going to draw the hydrogen gas vertically. There's going to be a connection between the oxygen and the hydrogen and the carbonyl carbon and the second hydrogen. And so since I'm depicting a skeletal structure, I'm not really obligated to write those hydrogens down. But if I wanted to write those hydrogens down just to show my work, I can. And so the reason why I'm writing it in pink is for you guys to see where that hydrogen is located. And then the other hydrogen that's in black is still going to be on that same carbon. Now, if you count the number of bonds around this primary carbon, I have 1, 2, 3, 4 bonds. And so that carbon still has its octet. And I do get a primary alcohol. Notice that the alcohol is at the very end, and there's only one connection to that central carbon. All right, so this last example, same idea. We're going to treat this with H2PD. Notice that this is a aldehyde. And so if we have an aldehyde, we're going to go ahead and convert that into a primary alcohol if we treat it with H2PD. So this is a reduction process. So I'm going to go ahead and copy the basic frame of my parent chain. Okay, so make sure that you guys are copying it correctly, right? So if you guys need to number those carbons, please do. So I'm used to it. I can kind of eyeball these things, but if you're not used to it, then I would number your carbons to make sure that everything matches up. All right, so since I'm going to produce a primary alcohol, this connection is from that last line connects to the original hydrogen that was on the aldehyde before the reduction reaction occurred. And so wherever that oxygen was, it now gets converted into a hydrogen. I'm sorry, not a hydrogen, but a hydroxyl group. And notice that there's a double bond here. When we treat double bonds with hydrogen gas and palladium, it becomes an alkane. And so it turns out like this double bond, I'm sorry, not this double bond, this H2PD reduces both the aldehyde and the alkene at the same time. All right, and so that's pretty much it for this reaction. Hopefully you guys understand my train of thought. If you guys have any questions, just go ahead and email me. The next chapter that we're going to cover is chapter 13, which is carbohydrates. And so I'll see you guys when I see you. Bye.